Well, good afternoon, everyone, and welcome. I'm glad to have all of you here. It looks like we've got a great participation for our talk today. Looks like several hundred people have joined us today for this inaugural research address for the CU ANSCHUTZ Medical Campus. So at the top, I just want to take a few moments to thank everybody who helped to prepare this talk. It took an alarmingly large number of people to pull this all together, and I'm grateful to each of you for your help. The agenda today is shown here. So I think if there's a theme to this talk, and really a theme to both my career and how I look at research, it's progress through collaboration. I think we have a wonderful collaborative environment here on our campus. I'm proud of that. I think many of us are proud of the environment we have here. I think there's additional chances for opportunities for collaboration, both within schools, between schools, and extending out to other campuses. And that should be something that we think about. If you look at the research work we're most proud of, some of it which we'll highlight today, and some of which we're proud of in our own personal careers, it's largely through significant collaborations, and really the more diverse and diversion of fields when they come together for collaboration, the bigger the impact. I'll give a brief welcome and a bit of background. We'll talk about Central Research Administration and really what that means and what that represents. We'll also touch on some of the metrics of research on our campus, both sort of metrics of research as well as the finances and our sponsored research portfolio. We'll then pivot to talk about our response as a research community to COVID. It's been impressive and frankly heartwarming, and we have some great examples to represent that work. We'll then pivot to the richness of our research community in general. It's striking the amount and the diversity of different research that we do on campus. Last, we'll talk about some special projects and initiatives that we have underway. So just a little history of myself. I started in this role in March of 2020, March 1st. It was an interesting time to start a new job in this setting. And as we all know, there was a dramatic change with COVID-19 that affected research just like everything else in the world. So this introduction and the outreach I intended to do was really delayed. So I'm originally from Minnesota. I went to the University of Minnesota and received my medical degree from that institution. Very fortunate to come out here to Colorado back in 2003 as an oncology fellow. And I did work at the 9th Avenue campus. So those of us that remember the BRB, the basic research building at that campus. In 2006, I decided I wanted to pursue a career in academics. And so there's some interesting things happening at the ANSCHUTZ campus. I remember distinctly that decision to say, let's start here and see where this goes. And it's only gone really up in terms of this institution and what we've done. My research is primarily clinical and translational. I've served as a principal investigator on dozens of trials here at the CU ANSCHUTZ Medical Campus, ranging from phase 1 to phase 3. I've led a couple of NCI-sponsored national trials through SWOG. We have a translational lab that's been focused on therapeutic development, biomarker development in urologic cancer, primarily prostate and bladder cancer. We have current funding through a DoD project looking at high-dose testosterone in prostate cancer. Collaborate with Steve Nordine on that project. We've been thinking about that for years, and it's great to be able to pursue that work. We're also currently collaborating with Jared Brown in the School of Pharmacy. Jared's leading a project looking at novel therapies for bladder cancer. We also have a long-term collaboration with Juan Park at CU Boulder. Juan and I have been partnering for many, many years. We have a patent together and are looking at an SBIR grant. Should also mention that we had a faculty startup a number of years ago and have received a number of SBIR grants. So that's my own research portfolio and what I have done in the past. In 2012, I transitioned to more of an administrative role. I don't know if that was necessarily the plan at the time, but that's the way things worked out. There was an opening as the medical director of the Cancer Center Trial Office. So I served as the medical director there for a number of years. That's a unit with about 100 personnel and sponsors essentially the cancer clinical trials and supports hundreds of accruals every year to those trials. 2015, I served as the associate director for the Cancer Center. We worked on the submission of the Cancer Center Support Grant, which was a very educational experience. In 2016, I served as the chief clinical research officer for UC Health and as the associate dean for clinical research. And then in August of 2019, I served as the interim vice chancellor for research, and as I mentioned, in March of this year, I transitioned over to the role of vice chancellor. So that's just a bit about myself. I feel very fortunate to be in this position. I think there's a lot of things we can do together. There's one question I received more than anything else since I started. how do you fit? What does that role do? How do you work with schools and the university and our hospital partners? So I want to just take a few minutes to talk about what is the Vice Chancellor for Research. And you might have noticed we've talked about ourselves differently sometimes. In the left lower corner, you see university research. We see ourselves in this unit, and this is the big we, it's a broad unit of people, as serving the needs of researchers across campus in a variety of realms, from lab research to clinical research to community to outcomes to big data, all those sort of things. So we are the infrastructure that supports that. We have a diverse and large research community. I pulled a couple of numbers. I like numbers, so there's probably too many here, but hopefully you find it useful and insightful in some ways. There are 5,000 faculty members on campus. It's just a little bit north of 5,000. There are 1,500 PRAs. There are 300 postdoctorates. EH&S oversees in excess of 2 million square feet of research space. We have over 2,000 open clinical trials at any given time. And Amy Gannon tells me over 5,000 sponsored research projects last year. So it's a large and broad research portfolio. If you think of the pillars of our Office of Research Support, I'd like to point out 3 of those main pillars. One is Allison Lakin, regulatory compliance. Another is Jory Lazinski in the OLAR division, as well as Amy Gannon, who has a lot of important financial responsibilities, including grants and contracts. These are the 3 main pillars of how we provide support to the institution, individual researchers. Now, this slide can become a little bit of alphabet soup. For those of us that are involved in these different committees, we recognize what all these things do. When you put it on a piece of paper and you put it on a single slide, you recognize just the broadness of this level of support and the infrastructure that's necessary for all the work we want to do, whether it's Institutional Biosafety Committee, the IRB, OLAR, anything else we're doing. All these are fundamental. And I'll tell you, having become even better acquainted with the team that supports these different functions, they are a highly dedicated group. There's several hundred people working in this particular unit, and they're highly dedicated to promoting and allowing and the research that all of us are doing. And it's really a wonderful group and team supporting it. They do a lot of things to support our research. Another thing that's important, I guess, in this office we should mention is research integrity. It doesn't come up very often, but it's very important that we have integrity in the work we do. This group also maintains, of course, the accreditations that are fundamental to receiving grants. So as I mentioned, I'm a clinical and translational researcher. I have a lab, but it's not a fundamental basic science lab. It was clear to me from the very beginning it was important to have a strong voice and advocacy for basic science research within our group and office. We were frankly a bit overwhelmed by the applicants to this job. We had many stellar and qualified candidates. It made the selection very difficult. I was also very delighted that Lori stepped forward and accepted our offer to start the position as Associate Vice Chancellor for Basic Science Research in October of this year. Lori has immediately made positive impact on our group, both in her ideas and her energy, and it's been great to invite her to the team. She's, of course, a professor of pediatrics and cell and developmental biology. She's internationally recognized for her work and research in diabetes. She's had many significant leadership roles, including as director of the Diabetes Research Center and the Barbara Davis Center. She serves as director of basic and translational research. She also holds the Sissel and Finlow family chairs. So we're delighted to have Lori in this role and as part of our team. Okay, so just a brief look at the leadership team and a broad view of the research community. So again, I told you I like numbers. We're going to put a few up here. So if you look at just the volume of what we're doing, again, it can be impressive when you think about it and put it down on paper. So the Office of Grants and Contracts looks over 3,000 proposals and grants every year. That equates to about a dozen a day. There's 1,600 or so research contracts that are coming in from other entities, at least another 1,100 that we are sending out. So I mean, that's over 100 documents that they're managing for us in support of our research every week, which is impressive. If you think of the clinical research and IRB support level, the Zen desk, as it's called, the help desk, has over 17,000 contacts from us requesting help that they respond to annually. There are 25 courses offered, which provides education to over 800 attendees. In terms of our FDA regulation, this has really improved over the last couple of years with some dedicated people helping us interact with the FDA for IND and IDE, for special permission to use investigational agents. There are 94 of those now underway. And this number I had to check a couple of times. It was a little bit north of where I thought it was. The IRB has over 5,700 active projects with approval at this time. And again, there's a specific definition for clinical trials, which constitutes over 2,000 of those. I wanted just to take a moment to talk about the IRB. So if we think about the types of protocols they look at, there's exempt protocols. So think of secondary research and surveys. There's full board, then there's expedited. John Heldens and his team over the last several years, and I've been able to watch him actually go through this process, identified secondary research, those exempt protocols. He changed the application, changed the process for reviewing those, and the time to review those applications of the IRB to COMIRB have gone from over 30 days to now just a handful of days, under a week, and frankly, 3 to 4 days on average. A very determined and specific effort to do that. If you look at full board applications, which by their nature require having a board and discussion and deliberation, even with that requirement, the time it takes to review those has gone from over 40 days to less than 30. It's a great example of the sort of process improvement that we can pursue and achieve together. Environmental health and safety. So I'm going to talk about them a bit during the COVID portion of our talk today. They do have a day job. It's a pretty busy day job. And a lot of us in the laboratories recognize what they're doing on a day-to-day basis. So for example, their team has over 500 laboratory audits every year. These are critical to our compliance, our safety, and also serve an important educational role. I know the audits we've had over the years, we've certainly improved our laboratory processes around that. If you just think of the sheer volume of, for example, regulated medical waste that we produce a year, over 100 tons. Again, EH&S is there to, in a very compliant way, deal with all of these issues and handle what we produce in research. We can also look at the OLAR group, who does great work. I receive a lot of compliments for Jory and her team, what they do. Their team is over 95 individuals. It's a very large team supporting this effort. They have over 100,000 square feet of current space. And with construction that's underway, that'll increase to 130,000. They support over 300 investigators, pursuing over 500 specific protocols. Again, a very significant operation. So thank you for indulging me. Let me run through some numbers. I hope you find that helpful. Again, it just speaks to the breadth of what we're doing here and the community that's behind you and supporting your work. Well, let's shift more to sort of the traditional grants and where we are as a campus. What we've done here is we've sort of looked across all the schools, across our campus, what does the research portfolio look like? And so if you go back over the last 5 years, you can see that our sponsored research portfolio 5 years ago was about $450 million. You can see over that length of time, over the last 5 years, this has increased to nearly $600 million this year. You can also see that we are fairly dependent on federal grants. I think that's a positive thing, that we are competitive in a very competitive sphere, but the majority of our grant funding is from the federal government. Another roughly $100 million comes from industry contracts and trials and so forth, and then foundations and associations beyond that. So again, what you can see here is a predominance of federal research and growth over time. We could also see here the breakdown of our funding by school. And you can see that the School of Medicine is the majority of the funding that we receive on campus, followed by School of Medicine institutes and centers. But you can also see, as we highlight here, the contribution of each of the different schools to this overall portfolio. We talk about School of Medicine institutes and centers, that speaks to the Barbara Davis Center and the Cancer Center. When we see the CU ANSCHUTZ centers and institutes, that speaks to the CCTSI and other groups such as that. This slide really talks about the proportion of federal funding in our overall portfolio. And one thing I'd like to point out here is if you look back 4 to 5 years, we had 55 to 35% support for these proportion of federal grants. So you can see that over time, not only has the overall portfolio increased, so again, reaching now close to $600 million, 599 to be specific, but the proportion of federal funding has actually kept up with this growth that actually is higher now. So we're at 58% the last year. I think that's a very healthy thing. One area that might cause us just a slight bit of pause is the idea that of the federal funding we get, as a academic medical campus, it's largely, in fact, the vast majority is from the NIH. This is fitting, it's understanding, but at the same time, it's likely an area where we could show some additional growth and increases. So for example, DOD, I mentioned we currently have funding through the DOD. Groups such as Dr. Vic Babarda's group is doing great work and trying to increase DoD funding. I think there's an opportunity for us to diversify our federal funding portfolio. Frank, if you look at partnership, we're going to talk about things we're doing to outreach. Obviously, we have a long-term connection to CU Denver. We also are doing additional efforts to increase the research at CU Boulder. They have a different portfolio of federal funding. I think through more collaborations and partnerships with them, we'll diversify this area. Okay, so we've talked a little bit about the numbers. We've talked about our organization and really what we are. I want to now do a pivot to what's the obvious story of the year for research and everything else, and that's COVID-19. So obviously, over the last 9 months now, we've seen dramatic changes initially and ongoing to how we handle research and how we sort of continue to do our mission in the time of a COVID pandemic. We put together a bit of a timeline. So it was March 16th, we moved to critical access only due to COVID-19, this campus. We very quickly started to establish working groups for laboratory-based research and community and clinical research. And I appreciate all those people that were involved in those meetings. By the later part of April, we had announced a laboratory research reconstitution plan. It was about a 10-page document. It took many hours of work and contribution from many on this call to put that together. By May, we had developed the same thing for clinical research. And by mid-May, we had started to activate that reconstitution plan. The overall situation had improved. We learned a lot. This novel virus wasn't so novel, and we knew what to expect to a better phase. We eventually moved to 50% and beyond 50% approval to come to campus, always keeping our census in a given time well below 50%. And in July, the community research framework was finalized as well. We reached a period of some homeostasis, right? There was a plateauing there during the summer that was calm. But of course, again, things have frankly deteriorated again clinically in the public health sense with the virus. I'll note that November 20th, we moved back to level red. And we just also note that we have learned a lot, and the way we can handle this situation now compared to March is markedly different. And that's due in large measure to your efforts and your help. So there's a couple themes you take away from the talk today. One would be, I guess, this idea that progress through collaboration is one of our real opportunities to think about that, to focus on that and grow that. The other one, another one I want to make sure you have is gratitude. We've gone through a tumultuous few months, to be sure. There have been a lot of changes that have happened suddenly, externally to us and everything else. And so I want to share our gratitude to all of you for your willingness to work through us during this time. We have prioritized safety and maintaining our mission. Those have been our priorities, and you've helped us along that road. I think we've accomplished both of those in a very thoughtful way. The research community has shown great adaptation and flexibility. It's been necessary. It's been generously given. I've seen develop a communal spirit of purpose and teamwork, perhaps in ways we haven't seen before. I think there were some voices that said it would be difficult to maintain the changes. Once we sort of open up the labs to a limited extent, things would change back to the way they were, and they haven't. They really, frankly, haven't. There have been specific incidents, but as a whole, we've continued to keep that vigilance and focus. I want to thank you for that, and we're not out of the woods just yet. There are a lot of groups we could highlight and talk about the contributions they made during COVID. I think none of those would be perhaps more notable than EH&S. And so we've listed here Ethan Carter and his leadership, as well as his managers, and really the whole unit of over 25 full-time staff, and frankly, many additional part-time staff. They've shown a great teamwork and leadership during this time. They established and maintained the COVID health screening checkpoints. The number there is, since May, we estimate there have been over 370 check-ins at those individual points. Now, again, the same person might be checking in every day. That's a remarkable number. That number, I believe, has helped to maintain our culture of vigilance. I mean, this process has helped to maintain that culture of vigilance. It's certainly been important to external reviewers that are looking at us and assessing our processes. They've developed new biosafety guidelines in response to the pandemic. At your request, Marcy and others have done a great job in adapting to this new information. They've added infection prevention specialist and Olivia, who's been absolutely fantastic. We've added many additional, I think now numbering around 15 additional contact tracers. So from the start of this, we've added additional staff significantly, which has allowed us to now conduct contact tracing on 1,600 individuals. That means when we do get a self-report or have an incident, we can do our own contact tracing in coordination with the public health authorities to very quickly address what's seen in this respect. We also have our own testing capacity. Cody Coburn herself, oftentimes here on the north side of the Fitzsimmons building, is doing symptomatic testing for those that either have symptoms or have a direct contact. We've also recently partnered to add asymptomatic testing on campus. It's been a wonderful effort. I just want to recognize that and how it's situated us for success. Other groups such as regulatory compliance have had a remarkable response. In June of this year, when the first drugs for COVID started to emerge, these investigational agents with a contract, the process that on a national level might take 3 to 4 months to complete, that is receiving a clinical trial packet and opening it to accrual, took 10 days. It was a very motivated sponsor. It was a very motivated team. And of course, the research team put in many long hours. I can attest to that. It's a remarkable achievement, which allowed patients to be treated within 10 days of us receiving that regulatory packet. They supported 30 emergency INDs that allowed investigative COVID treatments to be given to patients. They've coordinated the clinical trial prioritization with campus partners. Our partnership with both UC Health and the Children's Hospital of Colorado has been outstanding. In many ways, we've shown, I think, some of the highest levels of collaboration in developing our prioritization system together, and I'm very proud of that effort and thankful. I'd also like to talk a bit about the CCTSI, truly one of the real jewels of the research infrastructure on our campus. I've noted here Ron Sokol, who has very ably directed and led this group for many, many years. Very appreciative of his ongoing leadership. Would also say that during this time of pandemic, the CCTSI stepped forward, established a specialized research program to sponsor research in COVID. Here are the awardees for that project. And I think it shows a great partnership in adapting to this and sponsoring some really cutting edge research. Would also mention that there's a CCTSI town hall on the 15th. So if you haven't signed up for that, please go online and learn more about that. So we're going to take a brief break from my presentation. We've got about a 5-minute video we'd like to show now. I'm going to do my best to get that going.
Over the past 4 years, my lab has been developing high-throughput omics approaches to investigate the small molecule metabolite complement to the genome in blood and blood components. When the pandemic exploded, being sick from COVID myself at the beginning of March, we decided that our methods could be immediately applicable to investigate the impact of SARS-CoV-2 infection on COVID patients.
I am part of the multidisciplinary team of investigators that focus on performing multiple different assessments of the COVID patient samples to understand immune responses. Particularly, my team focus on understanding the immune cellular changes in the context of COVID infection. By understanding
immediately applicable to investigate the impact of SARS-CoV-2 infection on COVID patients.
I am part of the multidisciplinary team of investigators that focus on performing multiple different assessments of the COVID patient samples to understand immune responses. Particularly, my team focus on understanding the immune cellular changes in the context of COVID infection. By understanding what immune cell types change and how they respond to infection in the context of COVID, we can begin to formulate hypotheses as to what immune cell types we think are important in establishing a protective response.
I have the great fortune to have been the final link in a chain of coordination and collaboration that extended across campus to bring forward an antibody test for SARS-CoV-2 that was completely supply chain independent and based on the expertise and what we have on campus here. And together we came up and in the end in my lab brought to a clinical validation, a CAP-CLIA level assay that we could use to look for antibodies to SARS-CoV-2 independent of what else was happening in Colorado, what demands there were nationally or globally for those reagents, because we could make them here on campus and use our expertise.
We developed a dried blood spot-based assay so that people could collect their own blood samples for looking at antibodies, so that it requires not having a trained phlebotomist. And we validated that assay using this multiplex serology assay, and that assay is going to be really important because we can measure 2 different antibodies to SARS-CoV-2 in the plasma, and that allows us to distinguish between vaccine responses versus a natural infection in the community surveillance when the vaccine ultimately gets here.
One hallmark of the COVID pandemic here in the U.S. has been just the extreme variation in how it's played out across the country. And we've studied characteristics of communities that have made them more resilient, helping to slow the spread of the disease and also reducing the adverse health and economic outcomes, particularly for vulnerable and historically marginalized populations. And the key finding that we found here is that it's the strength of the networks that exist between medical care, public health, and social service organizations that really has been effective in helping certain communities be more resilient to the pandemic.
We are conducting a number of clinical trials looking at therapeutics for treating patients with COVID. And our hope is really to figure out effective therapies now. These are move-in ready treatments. So we're truly looking to find things that will prevent people from progressing to severe disease, saving lives, as well as those who do recover, helping with their recovery, make it better, faster, reduce the burden of symptoms, get them back to work and life as soon as possible.
My role in the COVID research effort has been to orchestrate clinical trials for COVID treatment and prevention here on the ANSCHUTZ medical campus. We have conducted studies of COVID treatment both in the inpatient and outpatient setting, including drugs such as remdesivir, as well as the Regeneron monoclonal antibodies.
This study was a great partnership with the Arapahoe County Office of Emergency Management, and who reached out to the campus to have help in understanding the seroprevalence of SARS-CoV-2 within the community and the first responders.
At the start of the COVID-19 pandemic, my laboratory quickly mobilized to establish a SARS-CoV-2 laboratory at BSL-3 to support COVID-19 research efforts here on the ANSCHUTZ medical campus. Our efforts during this COVID-19 pandemic have contributed to a number of different research manuscripts, as well as 25 or more grant applications for funding, 1 patent application, and the development and implementation of diagnostic assays that are currently being used here in clinical laboratories at the ANSCHUTZ medical campus.
Over the past 3 months, we had a chance to publish now 5 papers on the impact of COVID-19 on the serum metabolome and proteome, but also on the red blood cell proteome and metabolome. A paper on damage to red blood cells has been highlighted by the New York Times. Specifically in this very paper, we did find that damage to red blood cells can result in changes in red blood cell membrane components. And since red blood cells are devoid of protein synthesis capacity, they cannot replace damaged components while they circulate in the bloodstream.
We have lots of collaborators here on campus and across the country. On campus, we've been working closely with colleagues in the Department of Family Medicine here in the medical school, also with colleagues over on the Boulder campus in the Natural Hazard Center. And we've also got colleagues across the country, in particular for the Preparedness Index work at colleagues at UCLA School of Public Health and at New York University.
The collaborative effort that has been in place, the mobilization of our resources has been tremendous, and without this 360-degree support for this research, we would not have been able to make the progress that we've made so far.
We could not do these projects without the spirit of volunteerism and altruism that comes from the patients and the families that participate in these studies.
You know, to see a pandemic and to be able to step forward and do something is really gratifying. And I have to say, this collaboration and the strength of it is going to probably forever be a highlight of my career.
I hope you enjoyed that video. I think it's a great demonstration of really how we as a research community in our individual ways and our collective ways have stepped forward to contribute and help in this time of pandemic. I'd like to thank all the people that participated in the production of that. So going back a bit, at the start of this pandemic, another key decision we made along these lines was what to do about biobanking. Again, think back to March, limited PPE, extreme pressure on the clinical systems, providers having many additional demands and expectations, limits on how we could access the clinical floors. At the same time, we saw multiple applications coming into the IRB, essentially requesting access to these same tissues. So we had a number of groups that all obviously wanted tissues in the same patients. We made a decision then to have one single institutional effort to collect samples for COVID-19. It was a big decision, but it was based on the reality of the time and the limited number of patients we had. We worked together to set up a system. And so you can see we worked with both hospital partners, UC Health and Children's Hospital of Colorado. We looked at both prospective patients and patients accrued under a waiver of consent. We worked with the Biorepository Corps for coordination of this, with the Himser lab, which processed many of these samples. We used Encore for coding and connection of these samples, along with Health Data Compass for the clinical data. And importantly, we assigned a steering committee, or we appointed a steering committee. I'd like to recognize them here. In the early days, this, as I recall, we met at 7:30 in the morning, 2 or 3 times a week, regularly along these lines. And so I appreciate all the work that they did to essentially support these efforts, which were outstanding. And they gave great guidance to us as we worked through this. Right now, the biobank has thousands of samples available. And you can see that we've distributed about 3,500 samples through the biobank. Those go out to individual. So about 3,500 samples have been distributed to investigators across campus. There are thousands of additional samples. I'll move ahead one slide and show that there are many people we wish to thank for this effort. They're all listed here, and they're not all listed here, but many of them are listed here. There's many others. If you have questions about really these samples, there's a website. We direct you in this way. There's a REDCAP link to request the samples. And frankly, if you have any questions, reach out to Matt Steinbeis. One other decision, I'm gonna leave this up for a second. One other decision that we also made was to essentially look at the best way to utilize these samples. We saw many requests coming in early on this process where people wanted to run the same assays on the same samples. And so we said there must be a better way of doing this. So we thought, can we centrally collect the data that's being processed and share it? One great example of someone who's done this on campus is Joaquin Espinosa and the Linda Cernick Center with their Trisome Explorer. And I had a chance to see this previously, where Joaquin and his team had essentially put multi-omics data on the web and allowed access to this in a very positive and collaborative manner. So essentially the first question of Joaquin is, could we use this existing infrastructure and add in data as it came in? As these conversations continued, Joaquin found additional resources to do some of this central analysis. And so we brought in our samples with his efforts and the School of Medicine IT department, which helped us to put up this website. And we now have the COVIDOME.org. So you can note here, COVIDOME.org, which is a way to look at this multi-omics approach that we've taken to this problem. So again, the idea is that we've got roughly 100 to 150 prospective samples where Himser has in real time processed these. We can do cytokine analysis, we can do metabolomics, and a variety of other sort of evaluations there and consolidate them on one site. And so from that, here's a recent slide that Joaquin shared with some of us looking at defining biosignatures of seroconversion, where he's looking at the COVIDOME and the information's in there. I'm not particularly computer literate. You probably have seen demonstrations of that already today. I myself last night spent about 2 minutes. I went to COVIDOME, the COVIDOME Explorer. I looked at the effects of the COVID status. I essentially looked at IL-10. I said, I think I heard that was interesting from one of the talks I had heard previously on this. You can see demonstrated here, here's IL-10 on the scatter. You can see some of the analysis that's already being done around that. This took me literally 2 or 3 minutes. So for anybody interested in COVID-19 or this multi-omic approach, I'd encourage you to go to the website shown here again, covidome.org, at the end of this, and reach out to Joaquin if you've got any questions. It's a very large group of people that have participated in that effort. They're all shown on the COVIDOME website. So just shifting from that a bit, we've talked about who we are as a university research unit. We've talked about our response to COVID, which we're very proud of. We've really done great efforts there. And we're going to broaden it out just to talk about the richness of our research community in general. And I'll say, over the last several years, I've had some administrative roles. I've been able to participate in a lot of meetings, sat in the executive committee of the Cancer Center and the CCTSI. Thought I had a pretty good sense of what was going on campus. There is so much that I don't know. So many people doing neat research that it wasn't to my awareness. And I think that just speaks to the size of our group. What I've tried to do here is pick out a few vignettes to really highlight some of the different great research that's going on around campus. So in this case, I'd like to focus on the work done through this group from the Cancer Center, which spans different departments, medicine, biochemistry, to be specific, looking at the translation of bench to bedside. So there are many great examples on our campus of bench research that's been translated to the clinic. But I think this is really a sterling example which has had great clinical impact. So this investigative team is one of the first to look at regimens that directly target leukemic stem cells in acute myelogenous leukemia. They had several key publications in 2018, which led to an FDA approval of venetoclax in November of 2018. And again, the clinical impact of this has really been profound. I would say that their findings showed for the first time that a therapeutic intervention can eradicate leukemic stem cells in patients with AML by disrupting the metabolic machinery driving energy metabolism. This resulted in promising clinical activity in these patients who have historically had very bad outcomes. So again, I think it's a wonderful example of team science leading to a really specific patient benefit. I also had a chance to recently visit with David Cloutier and look at some of the work that he's done, in particular related to the ORO-OTO-CARDIAC syndrome. So David is recognized nationally for his expertise in craniofacial development and also related to G protein pathways and function. So researchers and clinicians at the Children's Hospital of Philadelphia reached out to him with an unusual syndrome. They had a clinical case that didn't fit the current understanding clinically. And based on David's expertise, he looked at this case and did a number of evaluations. So he recognized really a point mutation in the endothelium A receptor, which is a G protein coupled receptor and fits very well within his expertise. This is required, this receptor's activation for lower jaw and middle ear structures. This identified mutation prevented G protein association with the receptor and thus the loss of receptor function. Essentially, through this connection with the mechanism along with the clinical findings, they've defined a novel birth defect syndrome called ORO-OTO-CARDIAC syndrome. Really, I think, shows the synergy between clinical and bench findings to elucidate a new clinical entity. I'd also like to talk about the work done by Donna DeBella. She's a member of the Colorado School of Public Health and the LEAD Center, the Life Course Epidemiology, Adiposity, and Diabetes group. LEAD's mission is to help families live healthy lives without diabetes and their sequelae. They do this by conducting observational research across the lifespan and translating these findings. The work she's done with a longitudinal cohort of ethnically diverse pregnant women and their offspring has really been a hallmark of this work. This is a large study. There's over 1,400 individuals involved in this. This is, I think, a great example of the development of a data set, in this case, a longitudinal cohort, which has sprung all sorts of different collaborations and, frankly, grant mechanisms. Again, I think one of the best examples I've seen of this sort of work has led to collaborations with the School of Public Health, obviously, the School of Medicine, CU Boulder, and well beyond that. Next, I'd like to focus on the work of both Larry Allen and Dan Matlock. I've been watching this project for a while in my previous role at UC Health. They're working in accordance with CORDS and the School of Medicine. So this work is quite interesting. They received a grant from the American Heart Association to recruit 300 patients. It's basically a patient-centered educational tool to educate patients with congestive heart failure about the ideal medications that they should be taking. The idea is if you educate the patients, they can then talk to the provider and better inform the drugs that they're getting to start the conversation with the provider. To be successful in this endeavor quickly and with a larger and more diverse group of patients, they decided to pursue an enrollment strategy across UC Health, all 3 regions. So Larry and his team reached out, developed research collaborations, and then very successfully enrolled patients from across the Front Range of Colorado and beyond, frankly. This work was recently presented to the American Heart Association in November. You can see from the right side of my slide here, guideline-directed medical therapy was increased substantially from 30% to 49% with this approach. I think it's a great example of how we can do collaborative research across the health system. Next, I'd like to say a little bit about Laura Saba and her team in the School of Pharmacy. So Dr. Saba's lab has developed bioinformatic and statistical methods to explore the role of genetics, epigenetic and environmental variation on complex phenotypes, including addiction-related phenotypes. So this has led to a Center for Excellence in Omics, Systems Genetics, and the Addictome with collaborators at the University of Tennessee. So I think what she's really struck on is an area of real need. If we look at drug abuse and addiction right now, this is an ongoing need. Frankly, with much of our focus on COVID, there's been just a bit less on this, but it continues to be a very significant health and public health need. Her theme has really been high-end computational resources combined with bioinformatics and biostatistical collaborations. And this has led to a multitude of collaborations with the School of Public Health, the School of Pharmacy, School of Medicine, CU Boulder, and well beyond. It's a great example of forming a resource that people need and then being able to collaborate across a number of different entities. I'd also like to focus on this particular program, Colorado Medication Assistance Treatment Program for opioid use disorders. And just as we previously described on the previous slide, the opioid and painkiller epidemic in Colorado has been a significant issue, to be sure. And I'd say this represents a great academic community collaboration, which has led to an increase in medication for opioid use disorder providers. So this is a collaboration between the College of Nursing and the School of Pharmacy. It now serves 20 rural communities in Colorado, working with local providers on implementation of a medication assistance program. This was a collaboration with the state of Colorado and the Attorney General's office. Again, I think it's a great example of us working with our community members to have an impact. This has impacted over 600 patients. Okay, I'd like to focus now on what are our current projects and our future plans for research. I, like the rest of us, I think we all get a lot of emails every day. I get over 200. I know many of us, almost all of us, get that number. We used to send out a lot of individual research-related emails. That was one piece of feedback I got early on. We have made a very determined effort to look at our communication platforms and our strategies. There's 3 main things that we've started with. One are the research bulletins, which we launched in May. So you may have seen these in the first and third Tuesday of every month. What we've done is try to take maybe 20 individual emails that we would send out and consolidate them into a single mailing that comes out again twice a month. We've done this as best we can in a bullet type format. If that topic and headline looks familiar, you can click on it and get more information. And this covers a lot of things, news and events. If we find novel funding opportunities, we always have a little section on that part. There's also an important section on limited institutional grants. Now, for those not familiar, a foundation might say, hey, CU ANSCHUTZ, pick one nominee from your institution to bring forward for our consideration. So what we do then is we advertise that via this means. That's why it's important. Lori Sussel and I are looking at it right now, and she's leading an effort to essentially establish a formalized process around that, taking what we've been doing to date in a more formal structure, and then evaluate that, pick one nominee, and then forward that forward. Now, the way to find out about those, again, used to be a lot of individual emails. This would be a key venue, this communication, to see those. There are also some Vice Chancellor communiques. We've had many more than we expected of those with COVID. So I expect those will be relatively infrequent in the future related to research. We've also revamped our university research website. So I'd invite you to visit that site if you hadn't recently. A number of people have said, I found this website, you should make people aware of it. research.cuanschutz.edu. It's got resources for lab-based researchers, clinical, community, and beyond. Another group that I wanted to mention and talk about today is the CTOP program. This is a pretty specialized group that plays a very important function. As we've watched the Gates Biomanufacturing Facility ramp up their production of treatments, CAR T treatments for humans, both for external entities as well as ourselves, we're starting to treat people now on campus with CARs that are produced and really developed on our campus. The CTOP program serves as a critical liaison between the manufacturing, the university, and the clinical teams. This is led under the direction of Michael Veneris. I very much appreciate all of his leadership. The associate director is Ben Achalier, who's also done a great job in setting up this resource along with the rest of the team, which is listed here. For those of you that might have interest in the cell therapy area, I would ask you to reach out to Mike Veneris and discuss that. I'd also like to say a few words about the AB NEXUS program. When I started this, I did have a brief amount of time to be able to talk to people and say, what do you think our biggest opportunities are? And many of us thought that increasing and strengthening our research partnerships with CU Boulder would be a key opportunity. Early on in my time, I had a chance to visit with my counterpart, Terry Fees, in CU Boulder. We developed a great conversation relationship around strengthening our partnership. And one comment was made to me initially is that doing research within the CU system was just the same as doing it with any other institution in the country. We've made concerted efforts to change that, to really focus on the special relationship we should have with other campuses in CU. Again, we've got a very strong relationship with CU Denver through our long-term historical bonds. But I think there are many opportunities to increase research partnerships, which really have been modest between CU Anschutz and CU Boulder. I want to recognize and thank both Kathy Bradley and Amy Gannon, who led those initial discussions and represented our campus as we set up elements of this new partnership, which we've called AB NEXUS for Anschutz and Boulder. We have a research and support program. We have $3 million of funding through the generous support of Chancellor Elliman, the Chancellor DeStefano, and Boulder, as well as the CU President Mark Kennedy. With that, we've hired Diane Liddell, which I'll mention on the next slide. We've also set up a grant program, which just went through its first round. We also thought it was very important to say, if we're going to encourage more research, we want to make sure that the research runs as smoothly as possible. We looked for what we thought were gaps in making this process run smoothly. We have a master collaborative agreement, which was executed again with the help of Amy Gannon. There's an expedited joint review process. And this just isn't for the grant program, it's for all research that would happen between our two campuses. There's reduced form requirements. There's not additional F&A on the first 25,000 of directs for costs for inter-campus subcontracts, which is a change from before. So I'd like to thank all of those that were involved in this. As I mentioned, one component of this is both CU Anschutz and CU Boulder have hired and brought on board a research development partnership specialist. We're delighted to have Diane Liddell in that role. She's got a lot of history in our campus, in our community. I would strongly encourage you to reach out to Diane if you have any questions about AB NEXUS, about collaborations with Boulder. She has a counterpart, a strong relationship and connection with her counterpart in Boulder. We've had people come and say, hey, our unit or center or division has interest in working with this unit, center, division in Boulder. Dan can help to facilitate that, set up virtual meetings, connect with the partnership liaison in Boulder. So I'd ask you to think about that and to reach out. As I mentioned, we just recently went through the first round of funding. We will actually find a bit of additional funding for this first round to fund 8 projects. So 5 of these fit in the category of new collaborations. And I'd like to recognize Lori Sussel's leadership in this program. Again, hit the ground running, and we're glad to have her on board. You can see there's just a great variety of different collaborations as shown here through the collaborators and these different programs. There's also a second existing collaborations protocol. These are $125,000 awards for groups that have more existing relationships. And we hope that several of the new ones will develop into existing collaborations for additional support. As Chancellor Elliman recently mentioned on his State of the Campus, it's great to see the Anschutz Health Science Building developing. For those of us on campus to watch the physical space develop, it looks like a very collaborative space, I will say. If you look at the individuals and groups that are going to be in that building, as well as the layout, I think it's a great opportunity in that regard. I'd like to bring 2 things to your attention. One is that there'll be an investigational drug pharmacy. This is being done in collaboration with the School of Pharmacy on the first floor of this building. For those of us that have managed investigational drugs and investigator-initiated trials before, it would be great to have this resource available. We're very proud of that. Also, the CCTSI will be moving really from the Loprino space for their outpatient efforts into this space. It's a beautiful space. It'll be a wonderful place for our patients to come for clinical research through the CTRC. In the final moments, I have 2 final slides as we talk about the future and what's next. as I've said, I think we've given a lot of great examples of progress through collaboration. We're going to do whatever we can to encourage and support collaborations in a variety of ways. AB NEXUS is a good example, but there are many more. Again, I think we could have more collaborations at the school level, within the school, between the schools, and across campuses. We'll do everything we can to facilitate and foster that. It is wonderful to have Lori Sussel on board. She and I are already making plans for further support of program project grants and really team science. Expect to see some additional announcements and details about that in the coming months as we put that together. She'll continue to facilitate support for basic science endeavors, and we've got a variety of ideas. Again, you may hear more about that in coming months. We look forward to promoting diversity, equity, and inclusion in our research mission. With now the appointment of Regina Richards and the conversations we've started with her, I think there's wonderful opportunities to expand our efforts in this area. I look forward to working with her. Communications, we've touched on this a bit already. We're working very hard to consolidate our communications for you. Again, if you take another thing out of this talk, watch your email baskets in the first and third Tuesdays. We work very hard to have a consolidated, useful piece of information regarding research to you every 2 weeks. Organizationally, one thing that came out of COVID, obviously, is the development of COVID official structures for the lab, for the clinics, and for community research. I think we're going to look for a way, that's been such an effective mechanism for us, and it's been such a wonderful collaboration. Frankly, feedback coming to us, and I think messaging going out from us, we'll look for a way of continuing that structure. Allison Lake and I have been visiting about process improvement program and establishing more formality around that. There's a great example in the IRB I shared with you. There are so many processes that we manage. We're looking at ways to improve that and further refine and improve our research support services. And that could expand across really all of our service lines. Again, I think there's wonderful opportunities to further develop research with our hospital partners. We have very strong partnerships with both Children's Hospital of Colorado and UC Health. I gave the example of Larry Allen and the work he's done across the UC Health system. We're also very eagerly watching the development of a chief scientific officer recruitment on the children and the child health research enterprise side. We look forward to a variety of ways that we can collaborate and support those efforts. In all this, we want to promote innovation in all types of research. Well, with that, I'm going to stop for a second. We've got some great panelists. Many of the people I've been mentioning here, I'd invite them to join the conversation here. I believe Jory had a conflict today and couldn't join us. There might be some questions, and I think that Laura, Amy, and Allison have been kind of watching that with us. Kind of strange, I can see people again. I hope that went okay. There were no major glitches with that. I appreciate your attention. So to our distinguished panelists, any questions that you want to kind of bring forward from the room or from yourself?
Yeah, there's a question about the COVIDOME. Are samples still being collected for deposit into the biobank, and are they only collected from UH patients?
So a couple of great questions. So we got the biobank and the COVIDOME. I'll separate those out just a little bit. We did do a brief pause in collecting samples some months ago, but we are, I think, as of this day, actively recruiting and gathering samples. Now, the logistical difficulty of that is we're not able to get in the patient's room for research. There's a PP and everything else around that. We've developed a system which you can do this mostly electronically, including the consent. We are actively recruiting healthcare workers. One of the deficiencies of the deficits we had in the first round, we didn't have a lot of outpatient. We didn't have a lot of people that converted from negative to positive. This has been an active collaboration with both the adult hospital and our pediatric hospital. There are pediatric samples in the biobank and in the COVIDOME. Lori, did that cover most of it?
Yep.
Any other questions in that room there? I'm kind of still sharing the screen here. I can't see the questions very well. Well, maybe that's all we have. I would say that I was tempted to do a kind of a live video demonstration of the COVIDOME. I just didn't. It's very easy to use. I would encourage anybody that has any interest to go to the website covidome.org and facilitate and really look at that website. We have held back. We do have some samples that correlate with those that are in the COVIDOME. So if you said, well, IL-10 is interesting, Tom, but I really need to look at this in correlation to it, my question is more pertinent with that. We do have some additional samples which the steering committee would look at for additional studies. We'd actually ask that you contribute that data back to the COVIDOME. We can put it in there and then incorporate it what else we know.
There was a question in the chat about what do we know about the impact of COVID on all labs or other labs? What is the data on furloughs, firing, closing labs, etc.? more importantly, was there an impact on women, faculty, and staff? Because several publications have shown a significant negative impact on primary caregivers and women. So what are our numbers and are we looking into it?
Thanks for that question. I'll say a couple of things and maybe invite Amy Gannon to comment too, because I know she's looked at this. There's clearly been an impact. There's just no way of getting around it. I think it's remarkable that we've been able to actually get as much done as we could with all the different limitations. You know, one of the interesting things is we had some census data from a few years ago, a year ago. We could compare. Our numbers are down from where they were, but not down as far as I would have thought a while ago. And I think what many people are doing is they're working in the lab and taking the analysis work at home. But there has clearly been an impact. I think we're doing whatever we can to minimize the impact. So Amy, I know you did pull some grant data a number of months ago, and you broke that down by faculty rank and gender. Do you know, can you speak to that off the top of your head? Or I'll do my best if you prefer.
Sure. So I think what was interesting is the COVID officials asked us the same question, and we pulled the data for fiscal year from March to June. We actually saw an increase compared to the same time period in 2019 for both female and male. So we were seeing an increase at that time. I think it's something for us to keep monitoring, and we could do an update where we pull the data now in 2021.
Yeah, Amy, I really appreciate you doing that analysis. And I think you're right. It was probably now 2 months ago that we did that, or so it might be nice to refresh that. But I think the message for me was there's been a huge impact. And I think people have done remarkably well adapting to that. And we just need to continue to do what we can to support people across all different facets. So there is a question there about, is the video available to be shared? I believe that is the intention. It should be on our website relatively soon. I'm going to read the next one. We got just a few minutes left. Outside of AV NEXUS, how balanced currently is the NSHUTZ Boulder collaborative research in terms of indirect cost distribution? I don't know if I can answer that one. I'll say a couple of things that I can say. So if you look at the amount of research, so again, if CU Boulder and CU NSHUTZ are both in that 550 to 600, 500 to $600 million range, pretty comparable, there was a disproportionately small amount of research was happening between the campuses. Amy and I looked at this early on, and we could track some subcontracts and so forth. There's a huge opportunity to do more, and there's willingness. I think a lot of enthusiasm. Didn't mention that we had 74 full applications for the first round of AV NEXUS. That much exceeds anything that we expected, to be honest. I don't know if I can really say anything beyond that. We are tracking. The other thing is that let's say you submitted a grant to AV NEXUS, but it wasn't successfully funded. I still think that that could lead to external grant submissions and everything else. Part of what we're trying to do is to bring people together. Even those that don't get these internal grants, we hope that they can form successful collaborations. There's a little bit of time left here. I just want to invite Lori, Jory, and Amy to make any additional comments that you'd might like to make, if you have any observations. Otherwise, I think with that, I'm going to say that we're at about an hour. I don't want to go over an hour. There are many people, including those in the panel and beyond, who helped to put this slideshow together. Natasha Palmer was particularly helpful, and I want to pass on my gratitude to her. It's really exciting to be able to talk about our research community. There's a lot of seeds we're planting right now, and I look forward to a chance to update you probably in about a year how that's going. As always, feel free to reach out to us with your suggestions. Thanks for your attention today, and be well.